Hi, and welcome back to Research Reviews. Ever wondered how we figure out what all of our genes actually do? We've sequenced a mind-boggling number of genomes, but for the huge chunk of the proteins these genes code for, their function is still a complete mystery. It's like having a giant library where only a few of the books have titles. This is a real challenge in biology, creating what scientists call an annotation inequality. If a protein is similar to one we already know, great, we can guess its job. But what about the truly novel ones? They often get left in the dark. What if we could infer function not from similarity, but from how genes have evolved together? This is the principle of guilt by association. Genes whose products work together in a cell, say in the same pathway or complex, are often under similar evolutionary pressures and tend to evolve in concert. This shared history leaves detectable signals. And a new computational method called EcoWeaver tackles this by predicting gene functional associations directly from these coevolutionary signals. It aims to inject new information, especially for those mysterious, unstudied proteins. EcoWeaver doesn't rely on existing annotations or text mining, helping to overcome annotation bias. So how does it find these clues? EcoWeaver is like a super detective. It uses 12 different coevolutionary signals. These signals are grouped into 4 main categories. First, phylogenetic profiling, which looks at what species have which genes. If 2 genes are always present or absent together, they might be linked. Second, phylogenetic structure, which compares the evolutionary family trees of genes. Similar trees suggest a shared history. Third, gene organization. It checks out where the genes are located on the chromosome and how they're arranged. Sometimes genes that work together are clustered. And finally, sequence-level methods. They analyze tiny changes in the gene sequences themselves. If 2 proteins interact, a change in one might need a corresponding change in the other to keep things working smoothly. EcoWeaver takes phylogenetic gene trees and optional genomic data as input. It then calculates scores quantifying the strength of the coevolutionary signals for pairs of genes grouped using the 12 methods across these 4 groups. These scores are then combined using an ensemble machine learning model to provide a final prediction about functional relationships. The research shows that EcoWeaver is highly scalable, analyzing thousands of genomes and millions of genes, exceeding the scale of many previous analyses. On benchmark tests, EcoWeaver's ensemble predictions significantly outperform individual methods. It was able to accurately distinguish genes in the same protein complex from unrelated ones. What's really exciting is that EcoWeaver can predict connections currently missing in major databases like KEGG and String. And this is exciting because EcoWeaver can now generate new hypotheses. For instance, it predicted a connection between 2 human genes which wasn't listed in the major databases. Later, experimental evidence actually showed that one gene regulates the other. Sometimes, even when EcoWeaver's predictions seemed wrong, according to existing databases, a closer look revealed that it was just uncovering real biological links that were just missing or not fully understood. This shows its power to push research forward. It can also classify the strength of these associations into hierarchical levels, giving more nuanced insights than a simple yes or no. This tool is a big step in tackling that annotation inequality that we talked about, because it predicts functions directly from the sequence data without needing prior knowledge. It can shed light on those mysterious, unstudied proteins. And it's built to handle the massive datasets that we're generating today. EcoWeaver is implemented as part of the SynExtend package for R, making it more accessible to computational researchers. While it doesn't replace lab experiments, it's a powerful tool for generating new, testable hypotheses and guiding future research. As sequencing data keeps growing, tools like EcoWeaver are becoming indispensable for unlocking the functional secrets of the protein universe. Ultimately, EcoWeaver represents a significant step forward by combining diverse coevolutionary signals with machine learning to predict gene function on a massive scale, shining light on the dark corners of the protein universe. To dive deeper into the specifics, including details on the benchmarking and case studies, check out the full research review at Joshua Berkowitz US, where you'll also find links to the authors and the original publication. Until next time, I'm Joshua Berkowitz, and this has been Research Reviews.